Welcome to G Fun Facts Online. For centuries, we pictured the brain as a VIP lounge, an exclusive sanctuary sealed off from the body's chaotic immune system by a fortress called the blood-brain barrier. We thought this fortress kept the brain pristine and protected. But this idea is being completely overturned. It turns out the brain isn't isolated at all. It's in a constant, intricate conversation with the immune system, mediated by a unique team of cellular guardians living inside it. This field, called neuroimmunology, is revealing that this conversation can be the difference between a healthy brain and a brain ravaged by disease. Today, we're going deep inside this hidden world to understand how these guardian cells work, how they can turn against us, and how targeting them is offering new hope for treating conditions like Alzheimer's, Parkinson's, and multiple sclerosis. Let's start with the brain's defenses. The main line of security is the blood-brain barrier, or BBB. It's not a simple wall, but more like a high-security border crossing. The primary building blocks are special endothelial cells, which are fused together by protein links called tight junctions, acting like molecular rivets that stop almost anything from just wandering in from the bloodstream. Assisting them are cells called pericytes, which act like guards, controlling blood flow and access. And overseeing the whole operation are astrocytes, star-shaped cells that act like mission control, telling the barrier to stay strong and helping to shuttle nutrients in and waste out. Together, these parts form the neurovascular unit, a functional team dedicated to keeping the brain's environment perfectly stable. Beyond this main barrier, the brain is also bathed in cerebrospinal fluid, or CSF, which acts like a protective moat, providing cushioning and clearing waste. For a long time, a mystery remained. How does the brain take out its trash? The body has the lymphatic system for this, but scientists couldn't find one in the brain. Then came two revolutionary discoveries. The first is the glymphatic system. Discovered just over a decade ago, it's a brain-wide plumbing network. It uses the tunnels surrounding blood vessels to flush cerebrospinal fluid through the brain tissue, collecting metabolic waste like a garbage truck. This system is most active while we sleep, which is why a good night's rest is literally a deep clean for your brain. Dysfunction in this system is now linked to the buildup of toxic proteins in neurodegenerative diseases. The second discovery was even more stunning. Scientists found functional lymphatic vessels, the very system they thought was missing, in the meninges, the protective membranes wrapped around the brain. This was a direct drainage pipe connecting the brain to the immune system's command centers, the lymph nodes. It meant the immune system could constantly sample the fluid coming from the brain, monitoring it for signs of trouble. Suddenly, the brain wasn't a sealed fortress. It was a highly regulated, actively monitored kingdom with secret passages. Now let's meet the guardians who live inside this kingdom. These errant neurons. They're the glial cells, long thought to be mere structural glue. We now know they are the brain's active immune workforce. First up are the microglia. They are the brain's resident macrophages, its frontline police force and sanitation crew. Making up about 15% of our brain cells, their job in a healthy brain is vital housekeeping. They are constantly surveying their surroundings for trouble. They perform synaptic pruning, carefully trimming away weak or unneeded connections between neurons, which is essential for learning and memory. They also act as the cleanup crew, engulfing and digesting dead cells and debris to keep things tidy. But when microglia detect a threat, like a pathogen or a misfolded protein, they transform. They become activated. This isn't a simple on-off switch. It's a spectrum of responses. On one end, you have the M1, or classical activation. This is the aggressive, pro-inflammatory bad cop mode. M1 microglia release a storm of inflammatory molecules designed to neutralize invaders. This is great for a short-term fight, but if they get stuck in this mode, their chemical weapons become highly toxic to our own neurons. On the other end is the M2, or alternative activation. This is the good cop mode, focused on resolution and repair. M2 microglia release anti-inflammatory molecules, promote tissue healing, and clean up debris without causing collateral damage. The key to a healthy brain is the balance between these two. In chronic diseases, this balance is lost. The microglia get stuck in the destructive M1 state. Next are the astrocytes, the most abundant glial cells and the ultimate multitaskers. As we heard, they help manage the blood-brain barrier and provide nutrients to neurons. But they are also critical immune players. When they sense danger, they can become reactive. And just like microglia, this reactivity has two opposing sides. We have A1 astrocytes, which are neurotoxic. In a landmark study, researchers found that activated M1 microglia release a specific cocktail of inflammatory signals that basically order nearby astrocytes to become A1 killers. These A1 astrocytes stop doing their supportive jobs and instead start producing substances that are poisonous to neurons. Then we have the A2 astrocytes, which are neuroprotective. Triggered by different signals, they release factors that promote neuronal survival and repair. The interplay between microglia and astrocytes is a critical axis in brain disease. An M1 microglia can create an A1 astrocyte, and together they form a toxic duo, creating a vicious cycle of damage. What's even more incredible is a recent discovery called astrocyte immune memory. It turns out that astrocytes can remember a past inflammatory event. If they face a new threat, they mount a faster and stronger inflammatory response. Since astrocytes can live for years, this memory could explain how inflammatory conditions like multiple sclerosis become chronic and self-perpetuating. Other cells like oligodendrocytes, which make the myelin insulation for neurons, and ependymal cells, which line the fluid-filled ventricles, also participate in this complex immune dialogue, sensing and reacting to inflammation. So what happens when this carefully balanced system of guardians and barriers breaks down? This is where we see the roots of many devastating neurological diseases. Let's look at Alzheimer's disease. It's defined by plaques of a protein called amyloid beta and tangles of another protein called tau. For years, the focus was on getting rid of the amyloid, but now we know inflammation is a central driver of the disease. In the early stages, microglia try to clear the amyloid plaques, but as the plaques build up, the microglia become overwhelmed and dysfunctional. They get stuck in that pro-inflammatory M1 state, releasing chemicals that damage neurons. Even worse, their ability to eat amyloid gets impaired, so the plaques grow, which fuels more inflammation. It's a vicious cycle. Then, these M1 microglia activate nearby astrocytes, turning them into the toxic A1 phenotype. This toxic team-up of M1 microglia and A1 astrocytes is responsible for a huge amount of the neuronal death that causes cognitive decline. A similar story unfolds in Parkinson's disease. Here, the problem protein is alpha-synuclein, which forms clumps called Lewy bodies. This misfolded protein is a powerful trigger for microglia, switching them into the M1 state. These activated microglia release toxins that are especially deadly to the dopamine-producing neurons whose loss causes the symptoms of Parkinson's. The microglia can even spread the disease by failing to properly digest the alpha-synuclein and instead spitting it back out in a more toxic form that can infect neighboring neurons. Then there's multiple sclerosis, or MS, a classic autoimmune disease. In MS, the body's own immune cells from the periphery, like T cells and B cells, breach a compromised blood-brain barrier. Once inside, they launch a direct attack on the myelin sheath and the oligodendrocytes that make it. But they don't do it alone. They recruit the brain's resident guardians, the microglia and astrocytes, which amplify the inflammatory storm, contributing to the damage. Reactive astrocytes can also form a dense glial scar around the damage, which can physically block repair efforts. This new understanding, from the leaky barriers to the road guardians, might sound grim, but it's actually the source of immense hope, because if we understand the mechanism, we can design ways to fix it. This is revolutionizing the search for treatments. Instead of just trying to protect neurons, scientists are now targeting the immune system within the brain. One major strategy is to modulate the guardians themselves. Researchers are developing drugs that can flip the switch, pushing M1 microglia and A1 astrocytes back towards their protective M2 and A2 states. For example, therapies are in clinical trials that activate a receptor on microglia called TREM2, which boosts their ability to clear amyloid plaques and calms their inflammatory response. Another avenue is to restore the fortress. This includes therapies to seal a leaky blood-brain barrier or to boost the glymphatic system's waste clearance. Lifestyle changes like ensuring you get enough deep sleep are a powerful way to enhance this natural cleaning process. And we can't talk about immunity without mentioning the gut. The emerging science of the gut-brain axis shows that the trillions of microbes in our intestines have a profound effect on brain inflammation. An unhealthy gut can lead to systemic inflammation that compromises the blood-brain barrier and activates our brain's guardian cells. This provides a powerful link between diet and neurological health. For diseases like MS, therapies are becoming incredibly precise, moving beyond general immune suppression to targeted strikes against the specific B cells and T cells that cause the damage. The old idea of the brain as an isolated kingdom is gone. In its place is a far more exciting reality. A dynamic, interconnected ecosystem where immune guardians and physical barriers work in concert. When this system works, it maintains the health of our most precious organ. When it fails, the consequences are severe. By learning the language of this intricate neuroimmune dialogue, we are finally moving beyond treating symptoms and are starting to target the root causes of neurological disease. The future of brain health lies in restoring balance, calming the storm within, and empowering the brain's own guardians to once again protect and preserve the mind. Thank you for listening to G Fun Facts Online.